will be to review the background of influenza A viruses and pandemic viruses, and review the introduction of influenza A H1N1 PDM09, which is what was previously referred to as the swine flu when it was first introduced in 2009. We will look at the introduction of this virus into Jamaica. We'll look at the continued circulation of this virus in Jamaica. Was it here or not? Has it just reappeared? We will revisit the global influenza surveillance and response system and see how surveillance in Jamaica actually collaborates with this system. And we will also look at the current circulating seasonal and zoonotic influenza A viruses, and discuss a little bit about pandemic influenza preparedness and the status of immunization among Jamaican healthcare workers. Now we know that there's a lot of emphasis has been placed on the circulation of influenza, particularly from many years back, and we are well acquainted with the 1918 pandemic in which there was an influenza A H1N1 virus that actually caused the pandemic at this time, and it is believed that it was responsible for approximately 50 million deaths worldwide. In Jamaica alone, Jamaica was also affected by this virus, and it was introduced into Jamaica, and along with Guyana and Barbados, there approximately 10,000 deaths occurred at that time. Since the 1918 pandemic, there have been several other influenza A viruses that have caused epidemics, outbreaks, and pandemics. Included are other subtypes of influenza viruses, such as the influenza H2N2 viruses, H3N2, and as we said, a reemergence of the H1N1 PDM09, which I will explain a little bit later, is different, and it's not the same virus as the previously circulating seasonal H1N1 that was circulating prior to the pandemic in 2009, and that one is still a little bit different and was different from the 1918 pandemic virus. So although we refer to H1N1 viruses circulating now, swine viruses as initially it was called, PDM09, we have to try and remember that prior to this there were other H1N1 viruses that had circulated. So the importation of this novel influenza A virus, H1N1, occurred early in 2009, in about May, in epidemiological week 20, and it was associated with importation from a Jamaican who had traveled to New York and back. When they returned back, initially we had the importation of the cases, and the cases became sustained in their mode of transmission, and here we can see the distribution of the cases from May straight through to week 52 to the end of the year. So we see we had two waves or two peaks then. In total, we tested about a little over 4,800 samples, and these samples were distributed between severe cases and also non-severe cases. The severe cases we refer to as severe acute respiratory cases, and the non-severe ones we'll refer to as ARI, or acute respiratory infectious cases. Now, this virus that emerged in 2009 was different from the previous seasonal influenza A H1N1 virus circulating, in that the genetic components of it had components from the North American swine virus, the North American avian virus, and also the North American human and Eurasian swine viruses. So in referring to that, since 2009, the introduction in 2009, influenza A H1N1 PDM09 which was previously or initially called the swine flu, it did not disappear. We have been having sporadic cases, sometimes more than others, depending on the influenza season. So this virus has continued to circulate in Jamaica and also in the Caribbean at different levels and at different times, different seasons. Sometimes you may have had larger number of cases than others, but the virus, influenza virus, is such in that I'm going to refer a little bit now to the actual characteristics of the virus, but we're not going to go too much into the detail of the characteristics of the virus, because I want you to be able to understand beyond H1N1, because there is a bigger picture. The influenza virus is an RNA virus, and it is different from other viruses in that its RNA genome is segmented. It is divided into 8 segments. Because of this, the ability to mutate is increased, it's higher in influenza viruses than in influenza A viruses compared to other viruses. In addition, influenza A viruses do not only infect humans, they can infect other animals such as the swine, the avian species. The avian species, that's the main reservoir for the influenza A viruses. They can infect birds, swine, horses. So humans really become infected as aberrant hosts or bystanders. But what happens over the years is that the influenza virus circulating among humans adapted to humans, and therefore these are human, we term these the human seasonal influenza viruses. But we still have influenza A viruses that circulate primarily within the animal species and that are continually evolving. Now, when we have the interface occurring between humans and animals, between the influenza A, different types of influenza A viruses, there's a possibility that there can be some mixing of the genes, and this is where predisposes to what we call antigenic drift or shift of the virus with the emergence of new subtypes. The influenza A virus, just quickly speaking, has on its surface, we note H1 and also N1. It has the H1, these are glycoprotein projections on the surface of the virus. And there are 16 H or hemagglutinin glycoproteins and 9 neuraminidase, and they are numbered or named 1 to 9 and 1 to 16. And depending on the combination of these, this will determine how we name the influenza A virus. There are also influenza B viruses that circulate. These influenza B viruses, however, are not subtyped as the A viruses are. And there are influenza C viruses, and these do not usually cause major illnesses, and therefore we focus on the influenza A and on the influenza B viruses. But today's talk is primarily about influenza A. So because we know that influenza A causes ongoing outbreaks and epidemics and has caused pandemics, the World Health Organization from 1947 commenced the global influenza surveillance system initially, and it has expanded to have over 143 countries now participating in this global network. And this map here that shows you in green is showing you actually countries that do detection of influenza viruses through their National Influenza Centres and share these viruses with the World Health Organization collaborating centers so that the World Health Organization is able to assess and characterize these viruses on an ongoing basis so that we can know if there's any emergence occurring in the viruses, because we do not want to have another repeat of the scenario that we had in 1918. As of this map is showing you the general distribution of the different subtypes of influenza viruses currently circulating worldwide. And in the light blue is the H1N1 PDM09. So you can see the circulation of this virus. This is the Central America and the Caribbean here. And the larger portion of it is due to the H1N1 PDM09. The darker reddish color is influenza B, and the dark blue is the influenza A that's not subtyped, and influenza, I hope you can see it. Well, the general gist of it is that majority of the viruses circulating during this period are the influenza A, H1N1 PDM09. And this pie chart further shows you the distribution globally from August the 30th, 2015 to the 13th of February 2016. And this data is care of the Global Influenza Surveillance and Response System at the World Health Organization. So generally, there's 76% H1N1 PDM09 influenza A and 24% H3N2. Now, H3N2, that's the subtype of the influenza A, and the H1N1, the other subtype of influenza A. These are the two primary influenza viruses that have been circulating worldwide since 2009. And here we can see in a little bit more detail the distribution of the viruses or the detection of the influenza A PDM09 viruses between 2013 and 2016. So if you look at the trend, 2016 is in the red, that's right here, and 2014 is in it's the blue line, and you can see that in 2014 we had an increased circulation of PDM09 as well. It was a little bit, it was less in 2013, and in 2015 it was the lowest circulation that we had. So prior to the current circulation in 2016, you see in 2015 the circulation was much less. So how do we detect influenza viruses circulating in Jamaica? Since 1952, the virology laboratory, in collaboration with the Ministry of Health, signed a memorandum with the World Health Organization to have the National Influenza Centre established at the Department of Microbiology at the University of the West Indies. The role of the National Influenza Centre is to monitor the ongoing detection and changes of influenza viruses, and this is done in conjunction with the World Health Organization and their collaborating centers. In addition, how do we get our samples? The Ministry of Health has designated throughout the country 70 sentinel sites that collect samples from acute respiratory cases, and there are 6 hospitals that are designated as hospitals to collect more severe respiratory cases, and these are termed SARI sites, severe acute respiratory infection sentinel sites. So some of the SARI sites also serve to collect RI, samples from RI cases. These specimens are sent through the Ministries of Health National Public Health Lab on a weekly basis, and the National Public Health Lab will document and code these samples and then send them on to the National Influenza Centre where the tests are done. When the tests are done there, the reports go back to the National Public Health Lab, and also the results go to the Ministry of Health Surveillance Unit on a weekly basis, and reports are prepared. The Ministry of Health prepares a national report, that is issued by the Ministry of Health. That's their National Epidemiological Report. Reports are also sent to the Caribbean Epidemiological Centre, CARFA, it was previously corrected, now CARFA. Reports go weekly to the Pan American Health Organization and also to the platform, surveillance platform of the World Health Organization called FluNet. And now they have an additional platform called FluID, which will also be monitoring both the epidemiological and virological surveillance combined. So what did influenza surveillance look like in 2015? This graph is showing you it was very low. It started from 2014, and you can see here predominantly in this blue, it's the H3. This blue that you're seeing here is influenza A H3. So you can see in the latter part of the year was very quiet, and we really did not pick up very much either ARI or SARI cases referred. The referral rate was low, and the detection of viruses was concentrated mostly among the influenza H3. So we did not have any detection within the population for 2015. So as I explained to you, the virus changes because of its nature, and this happens in other countries worldwide as well. In green, we see the circulation of influenza B. So for 2015, we had primarily circulation of influenza A, H3, and also influenza B. And the genetic characterization of the influenza B, the lineage, was a mixture of both B Victoria and B Yamagata. For 2016, the 2016 season started later. Normally, I'll go back to the previous slide. Normally, the influenza season starts around week 40. But you can see here it did not, and this in the earlier part of 2015 is actually a carryover from the previous season. But here you can see we had a quiet season in December and the earlier months that preceded, and then we saw increased detection of respiratory cases, of which varying proportions were positive for influenza A PDM09. We also see slight carryover here of the influenza A H3. So there were only the detection of 2 such subtypes of the H3 for 2016 thus far. I mentioned earlier that the reports are sent to Pan American Health Organization, and there's a weekly bulletin that is put out. There's a link below on the slide, and I'd encourage all of you to follow the weekly bulletins issued because they are online, and all of Jamaica's information is also archived on this site, on this website, so that you can easily go there and see the distribution of influenza viruses over the years. I've only put years 13, 14, and 15 here, but you can see, and showed you previously 14, it's a bit small, but if you look at 2013, you can see 2013 had a mixture, had a different combination of subtypes compared to 2014, and there was PDM09 circulating in 2013. So a little bit more into the antigenic characterization of the influenza A H1 PDM09 viruses. The World Health Organization convened a meeting of the collaborating centers in February, and at this meeting they have 2 meetings each year in which they analyze and discuss the viruses that are shared with them from the different National Influenza Centres throughout the world. And these viruses that are shared with the collaborating centers are compared, and they are, and then a decision is made as to whether, what the the most relevant viruses circulating are within the population, and this impacts the composition, or how the composition of the influenza vaccine is chosen for the particular season that is coming, and for that season. And they also match it with the current circulating virus to see what the vaccine effectiveness is. So referring to the characterization of the H1N1 PDM09, we will see that almost all of the H1N1 PDM09 circulating in this current season were antigenically similar and closely related to the vaccine strain. That vaccine strain, A. California 7/2009, is actually the vaccine that was referred to as swine flu from the PDM09. I'm not sure. Can you still see my screen, Andre? Because I got a sign a while ago. Okay, I'll just continue. So, in addition to it being antigenically related to the vaccine strain that was recommended for this season, the sequence analysis also showed that the hemagglutinin gene indicated that there were 2 new emerging subclades within the 6B clade, that's 6B1 and 6B2. In most countries, the proportion of 6B1 viruses expanded very rapidly from October 2015 to become the predominant virus. However, in China, the 6B2 subclade viruses predominated. Viruses within clade 6B1 and 6B2 are currently not antigenically distinguishable from A. California 2009-like viruses, and both clades continue to evolve. They also looked at this meeting, as each year, at the circulating H3N2 viruses, and these viruses fell into phylogenetic clades 3C2 and 3C3. Viruses in subclade 3C2A predominated in all regions of the world. I see a hand going up. I am not sure if I think we will pause and answer questions after. I mentioned that the influenza viruses circulate among humans and also among animals. Now, when we're referring to the zoo, but there are times when they cross the species barrier, and particularly since 2003, we had the emergence of influenza A H5, the re-emergent in 2003. Now, viruses within the avian species are classified as those that are highly pathogenic. In other words, they have a very high fatal rate, can range from 60 to 80% or 100%, and also are low pathogenicity viruses, or low pathogenic avian influenza viruses. So influenza A/H5 is one of these viruses that is highly pathogenic, and it has circulated since 2003, and it continues to circulate. And for this season, from September the 22nd to February 2016, H5 infections were reported in Bangladesh, in China, and also detected in birds. There were human cases in Bangladesh and also cases in China. Apart from the influenza A/H5, initially it was influenza A/H5N1. There have been the emergence of other zoonotic infections of the H5 subtype, including H5N6. Other zoonotic influenza A viruses that are highly pathogenic include, when I say highly pathogenic, I'm referring to those that if they, right now, they do not have the ease of transmission between humans and animals, between animals and humans. But if they do mutate, there is a possibility. Therefore, there's very close surveillance on these viruses to ensure that they do not become easily transmissible among the human population. So we have influenza A with subtype H7N9 viruses also circulating presently. These have circulated since March 2013, and they continue to circulate. During this influenza season, 44 human cases of avian influenza H7N9 were reported to WHO. They were all from China, bringing the total number of cases to 721 with 268 deaths reported. In addition, we have yet another influenza A zoonotic virus that has infected humans, and this was first detected in 1998. That was the first human infection that was detected. There are not many of these cases, but it's another one that we have to watch and monitor. 6 human cases of H9N2 have been reported during this period, and fortunately there have been no fatalities so far with the H9N2. And these cases were all isolated from humans in China. One was associated with severe disease, although the underlying conditions also likely contributed to it. So surveillance actually is a foundation for pandemic preparedness. And in assessing whether a country is prepared for a pandemic, because we don't know when a highly pathogenic influenza virus is going to be introduced into any country. We don't know where it will come from, but we monitor and we know where these viruses are and what the potential is, and there are assessment tools that are continually being developed to assess the risk of pandemic potential. So there are performance indicators which countries should become acquainted with and also assess their ability to detect, to monitor, how they share their viruses with the global network, and also the sustainability. The sustainability is crucial because any lapse or break in the surveillance system is a risk for delayed detection of a pandemic virus. This slide is showing you the improvements across 4 critical functions for pandemic preparedness over the different regions. And you see with AMRO, this is the Americas, the pandemic preparedness in laboratory and surveillance being assessed at a little bit under 50%. So the vaccine that is recommended based on the analysis of the viruses that are submitted to the World Health Organization collaborating centers for this year, the vaccine is a quadrivalent or a trivalent vaccine. In other words, it has 2 A's and 1 B, or 2 A's and 2 B's. Therefore, when you get the vaccine, you're not only being covered for influenza A H1N9, you're also being covered for the other A that's circulating, the H3N2, and also the most frequent B that's circulating. And if you take the quadrivalent, you would be covered for both A's and B's. Now, closing and coming to this slide, this information, this data is taken from the World Health Organization, in which they assess the distribution of vaccine doses, influenza vaccines to countries, and this reflects the coverage in the Latin America and the Caribbean for 2013. I do not have the update for 2014 or 15. We see here that Mexico had 716 doses distributed, followed by Argentina, and then Costa Rica with 36,000, Ecuador, Honduras, Cuba had 13,000, Jamaica had 2,000, and then Dominica 342. The others, it may be a case where the countries are not submitting the data, but this is the data that is documented with the WHO. So in summary, influenza A H1N1 PDM09 has continued to circulate in Jamaica since 2009. The circulation of the types and subtypes of influenza viruses varies according to year and to season, and surveillance is necessary to detect ongoing changes in influenza viruses. And there is a need to improve the immunization coverage among healthcare workers in the population in Jamaica and in other Latin American and Caribbean countries. Okay, thank you. Questions?
Thank you so much, Dr. Jackson, for a very thorough presentation, and it's really reassuring to find out that we have a reliable surveillance system in place. We will reserve the questions for the end of the presentations, and we will now proceed to Professor Lina, whom I will introduce at this point. Born in Lyon, Professor Bruno Lina graduated as a medical doctor in 1990 and a PhD in 1996. He is Professor of Virology at the Université de Lyon, head of the Virology Lab at the Hospices Civils de Lyon, and he is Director of the National Influenza Centre in the south of France. He is also Director of the French National Reference Centre for Enteroviruses, and he's a scientific expert for the French Ministry of Health and has been working as well on short stints for the World Health Organization, for example. He has carried out expert evaluations for French Institutes of Health and Medical Research, as well as international bodies such as the National Institutes of Health and Hong Kong University. Professor Lina is also president of the European Society for Clinical Virology, and he is associate editor for the Journal of Clinical Virology. Professor Lina, we're so pleased to have you. We're looking forward to hearing your presentation.
Yeah, hello to everybody. Shall I switch on the webcam as well?
If you wish, that'd be good. As you please.
Yeah, I can. Hopefully it should work. Good. There you can see that it's dark here because it's night, but it's a pleasure being with you. Can you see my slides?
Yes, we can properly now.
Okay, excellent. So what I'm going to tell you is something that will be complementary to what Dr. Sandra Jackson already said, and to better understand the ongoing problem that you've been facing in Jamaica with H1N1. Actually, you really need to understand how the virus was built, and you have to remember the different problems we've been facing during the pandemic and after the pandemics, and to better understand the current situation. You have to remember what happened before to have a better disease management of what is happening right now. And then I will try to build on the current situation that you've been facing and try to answer a couple of questions whether this increase and upsurge of the cases you had in H1N1 was something that was peculiar, or if it was something that everybody should have expected, and actually that nothing really special happened to Jamaica. So I can't switch my slide. I don't know if you can do it for me. There you are. Okay, excellent. Again, like I said, you really need to go back to go back to what happens in the very early phases of the epidemic and the making of the virus to understand really what are the reasons for having such an impact of H1N1 infections in the different countries all over the world. Remember, this virus just emerged from Mexico in a very short period of time, because between the real emergence of the virus and the time when it was fully pandemic, meaning that it was all over the world, it took only 2 months. So this influenza virus, H1N1 PDM09, is highly adapted to humans and has the capacity to be transferred very rapidly from human to human through classical transmission of human viruses. And like it was said, this virus actually was a combination of 4 viruses that had different hosts. And one thing which is very important is that the hemagglutinin, which is the major antibody, the major epitope of the virus, was coming from swine virus, and this swine virus reassort mixed the gene segments with the different viruses, like it was said before, to make the pandemic virus. So the H1, which is here, was coming from this swine virus. And actually, when you look at the history of these viruses, if you go back into the past, we know now that this virus that was in the swine and that was after that acquired as a human component of the human new virus in 2001, was somehow related to the viruses that were circulating in 1918. And when we did some phylogenetic analysis and we did some comparison of the amino acids we had in the different hemagglutinins that were circulating and emerging in 1918, the one that emerged in 2009 and the one that we had before this emergence, actually they shared the same common ancestor, but they evolved differently. And this because the virus evolved in the swine population while this virus evolved in the human population. But at the end of the day, this swine virus was the provider of the hemagglutinin for the new virus emerging. And then when we look at the sequence of this hemagglutinin, what we can see is that there's actually some common ancestor that was circulating in human beings in the 40s and the 50s, which means that the people that were living at that time could manage to get an immune response that could protect against this swine virus. And you could really well document that, because when you were analyzing the cases of H1N1 at the very early stages of the pandemic, we could see in your region, in Central America and in the Dominican Republic, that there was this virus that was circulating pretty well. And when you look at the demographics of the patients that died eventually of this infection, the age group was really in the younger age. It was not the elderly people that was dying, because the elderly people was protected through the immunity they acquired when they were very young and when they were exposed to the viruses that were circulating between the 40s and the 50s. So this we have to keep in mind, because the immunity that we developed is long-term immunity. We have the capacity to acquire an immunity that will last and that can be called again if ever we're exposed to the same virus. And that is basically what happened in 2009. And when you look again in the ICU admission and the mortality that was measured after the pandemic and during the pandemic with H1N1, what we see is that there's a pretty high level of ICU admission in the age between 50 and 65, even more than in the more than 65, which is coherent with what I said about the immunity. But nevertheless, when you're old, you're exposed to severe cases when you're actually infected by the virus. But remember that ICU admission was really very high as compared to the other age groups for the 50 to 74 age groups. And the second thing that we learned during the pandemic is that the antivirals can be very helpful. Again, this is something that has been analyzed in Mexico. What they did actually is just they monitored the incidence of influenza over the months at the early stages of the pandemic, and they correlated that with the usage of antivirals and the case fatality rate that was observed. And what you can see is that actually the incidence tended to increase after August and was pretty low as compared to what it was in September and October in the early months of the pandemic. And at that time, there was a very large usage of antivirals, and the case fatality rate was pretty low. But when somehow this antiviral usage dropped down, what we did is there was an increase of the number of cases that we did see, and there was also an increase in the case fatality rate. And at the end of the day, what we collected during the pandemic was really clear that the antiviral treatment is really helpful to reduce the mortality associated to the H1N1 pandemic infection. So back to the current situation. That's the picture that was shown by Dr. Sandra Jackson, just to tell you that, first of all, the fact that H1N1 is circulating at a high level in this region is not something which is not seen in the other regions of the world. And you can see that especially in Southern Africa, in Africa, in Southern America, and in some of the other parts of the world, H1N1 is circulating very intensively. So it is not abnormal that there's a large number of H1N1 cases. When you look at the number of positive cases for influenza that you can see in this region, there's an increase in the recent weeks of the detection of the viruses, and indeed the majority of the viruses that are detected are H1N1 PDM viruses. And like it was observed, you had the feeling that maybe there might be a kind of worrisome situation in Jamaica because you have quite a significant number of pneumonia that is registered, you have a high level of hospitalization, and it seems that the increase of H1N1 was pretty high as compared to the level of viruses that you could have in the previous year. Is it normal or not? Well, actually, again, the fact that you have a large number of H1N1 doesn't mean that there's something going wrong, but the question that you can raise after that is, if you have a large number of cases, does it mean that the virus has changed and that there are antigen differences that would explain that the virus is spreading better and is changing, so it's spreading in different populations? Are there variance factors that have been revealed in the recent weeks because of the high incidence and the elevated number of cases that are admitted to the hospitals? Are there other factors involved in these infections, severe infection, or are there any other causes? So, first of all, what you really need to understand is that the viruses change, like it was said, a lot. And when they change, it's because they have to modify the region of the HA, which is the target of the immunoglobulin. These immunoglobulins are neutralizing antibody. They are really the antibodies that protects you against the infection. And what happens sometimes is that when the virus is mutating, this hemagglutinin changes, and the antibody is not able to recognize anymore the targets there. And hence, the virus is able to infect again the same individual because the HA has changed. And this is the continuous change that we do see in every human influenza virus. This has not been seen so far in the 2009 virus. And you can understand also that when you have an HA, which is represented here, and an antibody that is represented here, when the antibody is binding to the HA cannot really bind to anything else, and especially to the receptor. So that's the reason why these antibodies are really efficient and are really very good neutralizing antibody when they are binding this region close to the receptor binding site of DHA. So what we did actually is that we did a sequence of the virus, and that was touched by Dr. Sandra Jackson a little bit earlier, and we identified different genetic groups. And the major genetic groups that we detect in these weeks is the clade 6-2b. And when you look at the reference strain, which is the California 7 strain, which is here, you can see that there's a lot of little mutations that have been observed. And so there was a question about whether these viruses, which are circulating at a very high level now, may be antigenic variants as compared to the California strains. And you know now that the answer is no. And when you look at what we do now, it's drawing an antigenic cartography of the virus. You have the virus here, and if this is the reference strains, and each dot is a different strain that has been analyzed by the WHO Collaborative Center. And when all these strains are gathering around the reference strain, it means that there's no change, and that antigenically these viruses are exactly similar. And you have some peculiar viruses that are quite different, but these viruses have not been responsible for epidemics. All the epidemic viruses are located here. So it means that there's absolutely no antigenic change, and hence what the WHO says is that although some countries has reported a high level of hospitalization related to the H1N1, there's nothing special with that virus. And we know that it is due to the fact that this virus is causing quite a lot of severe cases naturally since it has emerged, but there's nothing that has been changed as compared to the previous years. And the second thing that they say is that since these viruses are antigenically identical that have been circulating before, then they are related to the vaccine virus, and the antigenically closely related viruses are perfectly well protected when you're vaccinated. The second thing they did is that they have an analysis on the susceptibility to the neuraminidase inhibitors to see whether these viruses may have acquired a resistance to the neuraminidase inhibitors. And actually, all the strains that have been analyzed so far, but 2, are susceptible to the neuraminidase inhibitors. So we know now, based on the experience that we have, that the early administration of these drugs can reduce the severity and reduce the complications related, and eventually the deaths also related to this infection. So when you have at-risk group, you should focus the use of antivirals in these at-risk group, and you should start to vaccinate them if possible to avoid the infection, because the vaccine is most likely very good. So there's no antigenic change. The virus is stable. It has not changed so far. So those that are immune remain immune, and those that have been vaccinated are now protected. But nevertheless, you have this upsurge. We have this upsurge of severe cases. So why is it the case? And what happens actually? What we know again from the past is that there are several groups that have not been identified before, but they are really at-risk groups for these infections with H1N1. And the pregnant woman is really an at-risk group that has been pointed at after the pandemic, and the severity of the cases there are very high. So you have to protect the woman by treating them with antivirals as soon as they are infected, if you can do that. The elderly, when they're infected, because sometimes they can be infected, are also at risk to develop pneumonia associated to H1N1. But you also have diabetes group, you also have the obese, you also have the asthma that can present severe cases. The use of antivirals is really something that is very efficient, and the use of hygiene preventive measures are also very helpful and can stop the spreading of the epidemic and can stop, take part of the control of small epidemics that can occur in the hospital settings. things as easy as hand washing with soap or hand washing with any kind of detergent is very helpful to prevent the transmission of the virus. And when you have somebody that is sick, you can put him in a room and organize some kind of social distanciation. There's no need for a grandpa or grandma to visit the kids when the kids are sick. They have to stay away from the source of infection. What we know also is that the vaccine, like I said, are pretty efficient. You know that the efficacy of the vaccine against influenza is at most about 70% efficient. So what we do in real time now is that we tend to measure the vaccine effectiveness during the epidemic, and that's the first paper that was released by the Canadian people. It's always them that do the first job. And what you see is that when you look at the at-risk groups, all the age groups, you have a vaccine efficacy of 72% against H1N1, and this efficacy is really good for all the age groups. So the vaccine is protective, and this is consistent with the fact that the strain has not changed. But remember, again, the age group that is between 50 and 65 are those that are most likely to be admitted to the hospital and to develop severe infection and sometimes to die. This is not a surprise. It is something that is expected and that is directly related to the fact that the H1N1 is really having a special focus on this age group. And again, remember that the early treatment with neuraminidase inhibitors is something which is really reducing the odds ratio of having a severe infection or mortality. So this is now crystal clear, and this is a way of handling the infection. So overall, and that is my last slide, nothing peculiar happened to Jamaica. It is just the fact that you've been facing an H1N1 PDM infection with a large number of cases, and that is because you didn't have it the year before. Vaccine, definitely, you should do something to be able to provide this to the fragile if possible. And again, I listed amongst the fragile the pregnant women, which are really a target group for H1N1. Early antiviral treatment is very efficient for the severe cases. You can reduce the mortality, and it is something that really should be taken into account when you have severe cases. And you have to keep in mind some of the specificities that have each of the influenza virus. Like I said, the H1N1 has a target group which is between 45 and 60. That is the age group which is targeted by the H1N1. When you look at H3 and 2, if you have an epidemic there, it's the over 65 that will be the target group. And that is there where you will see the mortality and the high number of admissions to the hospital. And when you have a B virus, the incidence is very high between the 5 and 15 years of age. The mortality is low, but there the impact is always very, very important. So that was my last line, and I'd be very happy to answer to the question if you have any. Thank you very much.
Thank you, Professor Lina. Thank you, Dr. Sandra Jackson Betty. Very informative presentations, and I think many questions that we've had in our minds, or many concerns or high levels of anxiety that we've had about influenza A, H1N1 have been clarified here today. What I want to ask first, Dr. Jackson Betty, in our surveillance program in Jamaica, do we have the capacity to differentiate the different types of strains that are circulating, or are we restricted to focusing on one or two strains only?
No, we have, thank you, Johan, for the question. No, we have the capacity. We have been subtyping the strains for quite a number of years now. So we do that, and we also do surveillance for the avian influenza viruses in humans as well. So we refer to the WHO collaborating center, as Prof. Lina said, for them to look at the global picture as to what exactly is circulating in the northern hemisphere and in the southern hemisphere. So our referral to CDC, which is the collaborating center that Jamaica uses, is to gather the antigenic characterization information from the regions of the Americas, so that we can use the appropriate vaccine for our area, for our region.
Thank you, Dr. Jackson. Prof. Lina, let me ask you, with regards to the stability of the influenza A H1N1 PDM09, it's remained antigenically stable. Do we anticipate any changes coming, or what sorts of pressures, for example, misuse of antivirals or something that's happening in the avian population, what kind of factors would influence whether this virus changes or not?
Sometime the virus will change and there will be an antigenic variant that will emerge. This is due to the fact that the virus is undergoing a large immune pressure, and if he wants to remain stable in the human population, he has to escape to the immune pressure. And the only way for him to escape this immune pressure is to develop variation, antigenic variation. So these antigenic variation will happen sometime. It is not the case today, but we know that some of the strains that have been detected in China may be the first link to an antigenic variant. The usage of the antivirals does not change anything in the antigenicity. It doesn't. It has no antigenic pressure because it's targeting the neuraminidase, and the neuraminidase is a minor antigen for the virus. And so the fact that you are using neuraminidase inhibitors will not promote the antigenic change. It's only the immune pressure.
I see. Thank you for that response. We do have a question from a participant, Judith Francis Bogle, who is asking, in light of the many viruses that are circulating, if there are any specific criteria for using antivirals, given the benefit of early administration of antivirals. So the use of antivirals, is that based on what strains are circulating?
No, the use of antivirals is based on the patient that you are facing. If you have a patient in front of you which is showing anything of severe infection, like dyspnea, for example, you have to treat it as soon as possible. And you don't have to wait for the confirmation for influenza infection. If you're in the middle of the epidemic and you have somebody that shows the clinical signs and symptoms that are really representative of a true influenza case, and you know that this patient is at risk to develop a severe infection because of his immunity, because of his status, because of the clinical symptoms he has, then you can start the treatment immediately. If ever you can do biology afterwards, and the biology tells you that it is not influenza, you just stop. It's not a problem. But if it is influenza, you will have a safe time, and the sooner you begin the treatment, the better it is, clearly.
Thank you. We have one more question regarding administration of vaccines for pregnant women.
Yes. The pregnant women have been identified as an at-risk group, which is really showing severe cases when they're infected. You can vaccinate the mother at any time of the pregnancy. You don't have to wait for the 3rd month. They can be vaccinated from the very early stage of the pregnancy. You have to keep in mind that if a pregnant woman is doing a severe influenza infection, it is the mother that is at risk. The children, usually after they're born, they're safe. But sometimes what we do see in Europe is that women that have been infected by the virus, we can deliver the baby, but the mother dies. So, and the risk for mortality in the pregnant woman is very, very high. I think it's something like 20-fold increase when they're infected with influenza. So it is really a target group.
Mm-hmm. And I do have one final question before we wrap up, at least from me. It's interesting how the age groups, the different age groups that are targeted by the different strains, we've been used to traditionally the elderly or persons over 65 generally being more susceptible to influenza and persons with certain comorbidities, but apart from the partial protection that persons over a certain age may have because of prior exposure to a similar H1N1 strain, are there real distinct target demographics between these viruses?
Within H1N1, there's absolutely no differences. Anytime we have an epidemic with H1N1, we can see that the mortality in the over 65 is really reduced as compared to what we have with the other influenza viruses when they are circulating. That is an experience that we've been facing during the pandemic, where the mortality was really low, and it was really low because the over 65 were not infected. And we also saw that in the next epidemic seasons when H1N1 was circulating in Europe, and there was limited mortality with the over 65. But when these patients are infected with H3N2 or are infected with the B virus, then the mortality is significantly higher than with H1N1. And you have to keep in mind that when you have a drifted H3N2, that is a virus which has acquired antigenic variation, which has usually the capacity of spreading pretty well, and then you may have a very high mortality in over 65. Then you have to protect these people. This mortality in over 65 is mainly seen in the very old age group, over 80. There, the mortality can be up to 5 to 10% of the patients infected.
Okay, thank you. I'll invite both speakers to make any final comments. Dr. Jackson, Prof. Lina, any final comments from you before we close?
I'm not sure. Can you hear me?
Yes, we can hear you.
Oh, okay. I just want to close by reinforcing what Prof. Lima has said and thanking him for an excellent presentation. The vaccination coverage, which I showed in the last slide, speaks for itself in the need, and that is just the healthcare worker group, although it's the number converts to 32% coverage. Subsequent study done actually resulted in 15% coverage among healthcare workers. So I would encourage healthcare workers to reconsider getting immunized with the influenza vaccine. And I think there was one question that came up with regards to the coverage of the vaccine. Remember that the vaccine is trivalent or quadrivalent. Therefore, it will cover the circulating viruses once there is an adequate match between. And the GISRI system ensures that, to their best, that a proper match is produced each year. Thank you. Thank all of the staff from the National Influenza Centre of Jamaica and all of the collaborators at PAHO, WHO, CDC, and CARPHA. And to you, Johan.
Thank you, Dr. Jackson.
Before you leave, there is a question. Are there any local facilities that offer the vaccine?
In Jamaica?
In Jamaica.
Yes, there are. Because of the present, I would call it scare or concern. Well, of the number of increased cases affecting the elderly and the number of hospitalized cases, the usage of vaccine, the uptake has increased recently. So I think the Ministry of Health is actually collaborating to get additional vaccines into the country. But there are private doctors and private hospitals. I think if you call the National Influenza Centre or you refer to the Ministry of Health, they will be able to let them know which outlets have the vaccine.
Thank you. Any final comments, Prof. Lina, before we go?
Yeah, just one thing to add on what was just said. I think that you have a pretty good system and a pretty good surveillance in Jamaica now, which means that you can have a clear picture of what is going on in terms of circulation of the respiratory virus and the influenza virus. Now you're in a position of being able to change something in the circulation of the virus through vaccination and through a better management of the cases. So if you can start with having a good vaccination campaign that will target some of the specific groups that you would identify, then the people will realize that we can do something against influenza and we're not just sitting and watching what happens.
Thank you so much. I'm really pleased to have had both of you and to have had such high quality presentations and the reassurance that you've given us about the circulating influenza viruses and what we need to be looking out for. Thank you both so much. Thanks to my team at Caribe Wellness, Andre Henry in Kingston, and to Anastasia Rambaran in Bridgetown, Barbados. Thank you both. Have a good evening. You can also email us questions, or you can use the hashtag CWC webinar or on Twitter at Caribe Wellness, and we'll try to respond to some of those questions. Thank you, and have a good evening.
Just before we go, you can also visit our YouTube page to find previous and current videos.
Excellent. Thank you, Andre.
Okay.
Good evening,
Thank
everyone.
you.
Yeah. Thank you. Goodbye.
Goodbye.
Bye.